All right, so today we're going to be talking about mutations, and specifically, we're going to be talking about gene mutations. So remember that a gene is a small piece of DNA. So we're going to be focused on the base pair sequences of DNA. This is important to know that it's different from a chromosomal mutation, and that is when there's a mutation with a large piece of DNA. So again, the mutations we're covering today are gene mutations. They happen in a very, very, very small piece of DNA. So, to recap, we know that proteins do a large amount of work in the cells. The ultimate goal of our cells are to make proteins. We know that proteins are made through a process called protein synthesis. The first step of protein synthesis is transcription. Transcription happens in the nucleus where DNA is going to synthesize mRNA. Once that mRNA is synthesized, the mRNA will leave the nucleus and go to the ribosomes, where it will then go through translation. The mRNA codons will complementary base pair map with the tRNA anticodons. The tRNA will then drop off its amino acid and it will attach it to the growing polypeptide chain with those peptide bonds, thereby growing our proteins. So, our proteins are made by many amino acids that are bonded together by peptide bonds. So mutations can happen through a few different ways. Some of them we actually have some genetic predispositions to, and some of it actually comes from environmental influences, such as UV radiation, for example. So if the DNA is mutated or changed, then the nucleotide sequence will be different. That's what's actually changing, the order of those bases. So if DNA is changed, that means that the mRNA that is transcribed will be different. And if the mRNA code is different, then the amino acids it's coding for will be different. If the amino acids are different, then the protein will be different. So you can see it's a cascading effect. So by changing the DNA, we can ultimately change what proteins are being made. Some mutations are neutral. Well, actually, many mutations are neutral. Very few of them are actually considered seriously harmful, though there are some that are considered harmful or even fatal. And some mutations can actually lead to advantages. And we'll talk more about that when we get into the unit of evolution, as we talk more about adaptations and natural selection and so on and so forth. But for the most part, many mutations are actually neutral, meaning that they pretty much have no impact on the expression of that particular gene or protein. If there is one that is harmful, it can cause a change in the protein structure or a specific gene activity. So, a mutation is the actual change in the order of that nucleotide sequence. So, usually, this can come from bad proofreading from that DNA polymerase. So, remember during both replication and transcription, polymerase is going to actually act as a proofreading tool. In this case, it did not work very well and it missed a mismatched base pair. There are actually two types of mutations that we're going to talk about today. So we have point mutations and frameshift mutations. A point mutation is a change in one nucleotide base, so only one. It's a specific point in a base pair sequence. So by changing only one nucleotide, one base pair, that will only change one amino acid. So, for example, when we look at this first example, we have CCC. Here we have mutated the third C to a G. So instead of CCC, we now have CCG. If you look at the codon chart, they both actually code for proline. So nothing actually happened there. So this point mutation was not harmful at all. But in this example, we have UUA, that was the original sequence that coded for leucine. When that A was mutated into a C, it then coded for phenylalanine. So here we can see with a point mutation, sometimes it has no effect at all, while other times it can actually code for a different amino acid. Sometimes just one change in an amino acid can have a devastating effect on that protein, while most of the times the protein will still go on as normal if it's only one change in one amino acid. So, an example of what a point mutation would look like if we were using a sentence in English. A normal sentence would be the dog bit the cat. If we substitute one base for another, that was not supposed to happen. In this case, we're going to substitute T for the R. The sentence now reads, the dog bit the car. It was not the original sentence, but it still makes sense and it's still workable. So we can see a point mutation is not always that bad. A frame shift mutation is very different. It is actually going to shift the reading frame in which the ribosome reads those base pairs. So remember that a ribosome is actually going to read in sets of threes. So those three bases make one codon. That codon is considered a reading frame. So in a frame shift mutation, we are shifting those codons down, which can completely mess up the entire protein. There are two types of frameshift mutations that you need to know about, and those are insertions and deletions. So insertions are when we insert or we add an extra nucleotide base that does not need to be there, and a deletion is when we remove or delete a nucleotide or base. So every amino acid after that specific addition or deletion, every amino acid after that will be completely different. So an example of an assertion, if we were to use a sentence in English. So again, we have the sentence, the dog bit the cat. That is the normal sentence. Here we have added the extra letter S, and we have inserted it right after the G. So now we can see it starts off the dog, which was normal, nothing changed there. But after the S, the entire reading frame completely changed after that. One thing that's really important to note here, we have a leftover base here. The ribosome cannot read anything that's not in a set of three. So if there is one base or two bases that are left over, no amino acid can be coded there. It can only read in sets of three. But we can see that the rest of this sentence is completely messed up. So every amino acid here would probably be different compared to the original. An example of a deletion in which a base is removed, again, we have the normal sentence, the dog bit the cat. We have now deleted the letter D. So now the word the, which was normal above, it stays the same. But everything after that D was deleted is completely different. And so that we can expect the amino acids to be completely different. And if all of these amino acids are different, that means that the entire protein is probably different as well. So frameshift mutations are considered significantly worse because every amino acid changes after that mutation, which means you're going to create a completely different type of protein. That can be really, really dangerous depending on the protein that's being made at a specific point in time. So, just to recap to make sure that we understand everything with these gene mutations that we talked about today. Number one asks, what is a mutation? We know that a mutation is a change in the DNA sequence. Number two asks, what is a point mutation? We know that a point mutation is when there's a change in one specific base or one specific nucleotide. An example of a point mutation would be a substitution mutation. Number three asks, what is a frame shift mutation? A frame shift mutation is any mutation that causes the reading frame or those codons to completely shift. The two types of frame shift mutations are additions or insertions and your deletions. Number four says that we need to decide if the examples below are a point or frame shift mutation. Looking at this first example, we have our normal mRNA and we now have our mutated mRNA. So you can pause this video now and take a second to figure out if this is a point mutation or a frameshift mutation. So this mutation is actually a point mutation. If we match up all the letters, we can see that the reading frame actually stays the same. So there's not an additional or one less base pair down on this end. And as we come across and we match them up and look to see what changed, we can see right here, the G was substituted for a C. So this could potentially have no change at all, or it could change one amino acid. This protein may not be affected at all. So this would be a point mutation. So looking at this second example, take a minute now to figure out what type of mutation this is. So this mutation is a frameshift mutation. So the big key here, how we know this, we can see that there's an empty slot right here. So we can tell that this entire reading frame shifted. So we have two left over. So the codons do not completely match all the way up. We have two that are left over here. We are missing one letter here from this final codon. And so we can see here as we're looking through the letters, this G was deleted and the rest of the reading frame was completely shifted over to the left. Number 5 asks, which is worse, a point or a frameshift mutation? So we know that a frameshift mutation is significantly worse. As seen in the examples, it will completely shift those codons, which will completely change every amino acid coded for after that. At least with a point mutation, all the other amino acids are still the same. Only one amino acid may possibly change. It's a possibility. With a frameshift, it is a guarantee that multiple amino acids will definitely be coded or will be changed. One important note about frameshift mutations: it is actually worse to have a frameshift mutation in the beginning of the sequence because, like we said, every single amino acid after that will be different. If we have a frameshift at the very end of the DNA sequence, it will not have as much of an impact. So just make sure to remember that key point. So that is the end of our notes for mutations. If you have any questions, make sure to just let me know during class.